What's up, Noah? I hope this works. I thought it'd be fun and helpful for me to kind of explain what we talked about tonight at the study session. Um, so she sent, I saw Dr. Townsend sent out a message. We're not going over anything with neuroplasticity, so anything we covered starting with these slides Wednesday. It's just running from pufferfish slides, the ones you printed me, from the pufferfish on, which is about the conduction of action potentials. Um, and mostly, like 99% of what we talked about tonight came from the set of slides about synaptic transmission and integration. So I have that pulled up here, and I'll follow along. And then she gave me a couple key, you'll see when I send you the pictures, a couple key questions to kind of look at and go over, like verbatim, I think what will be on the quiz, and some types of questions that'll be on there. So this, I don't know if you can see my cursor, but this stuff right here, bada bing, bada boom, is probably, oh, I wonder if I can, oh, my computer's touchscreen, so I can just do that. So baller. How do I exit? Oh, exit drawing. Okay, let's get cut right to the chase. So this structure, oh, I can go back to drawing. So this structure right here is part of the neuron, and this structure right here is either another neuron or a muscle cell, or other neuron or muscle cell, something of that sort. I think those are the only two. I should probably verify that. I should know that. But it's only that transmission for transmitting action potentials. So it starts within this system here, where the chemical synapses, there are, in this slide is it saying, actually I really don't know. Yeah, this is what I was talking about with the, I don't know. I actually don't know what the heck I'm saying. I took a lot of notes, but, so precursor, okay, this is what I've read. So the precursor to the action potential is getting the neurotransmitter out of these little balls. I don't know what they're called, and they're done. So this structure right here is the presynaptic membrane. It's important to know that. This is the postsynaptic membrane, and these little green dots here are chemically gated channels. That's something key we talked about tonight. Those are chemically gated channels, and those are activated by the neurotransmitters. Once those are activated, they reach a certain point on, exit drawing really quick. They reach a certain point on, go down to the next page, this graph here. I wish I could make that bigger. I can't. Boom. Right here. This graph we looked at a lot, talking about how it reaches action potentials, because this is like what is happening when the neurotransmitter reaches the chemically gated channel. So when the neurotransmitter, little pink dot, leaves its little bubble because of the calcium channels. So I have true written to it next. So I think there's going to be a true-false question about that. So calcium channel is a voltage gated channel. It says true. I'll send you a picture of everything I'm reading. So also she said calcium the purpose is to move, fuse, and burst. That comes from the male end of the neurotransmitter reaching the postsynaptic membrane. Dude, this is so hard. How does it move across the synaptic? I have this written down too. I think it's in the notes on the slides. How does it move across? It moves via diffusion. So once those, I don't really understand that process so much. I might put a post in the discussion board thing on there. But once the neurotransmitter leaves the little balls on the first slide, if we go back up one, once they leave this little ball here, what's it called? This is when, I have one right here. So once they bind to this green box, the neurotransmitters leave, then they move across the presynaptic membrane. They diffuse through the synaptic cleft here, which is the gap in between them. They go over to the green box, right? They go over to this green box, which is our chemically gated channel, and that is activated chemically. So the neurotransmitter goes to this one. There's only 1-to-1. I think like it's only 1-to-1. Once those are reached and opened, that is when we get to our reaction here. Now this reaction is key that we know the details of. So stimulus, this is the binding of the neurotransmitter to the thing, to the neurotransmitter to the chemically gated channel. That is our stimulus that brings us to threshold here. Once threshold is reached, there is other channels that are not in this diagram. Now I'm like, why wouldn't this be in the diagram? Because we're expected to know it. It makes no sense. But there's other receptors here that'll go off. Once this is chemically activated via the neurotransmitter, there's voltage gated channels that will open up. That brings us to our, so then once those voltage gated ones are open, so negative 70 millivolts is resting membrane potential right here, it says. That's resting. And so a little bit of stimulus brings it up to once it reaches negative 55. Anything, so EPSP and IPSPs, as I understand them when I talk to her about it, are just a means of, I have it written down here, are called graded potentials. And are almost, this is a true-false question. So true-false, it's in the notes, I'll send you a picture. It says graded potentials are almost always caused by opening of chemically gated channels. That statement is probably going to be on the test, almost verbatim. So graded potentials, so the EPSPs and IPSPs, are almost always caused by opening of chemically gated channels. So once the neurotransmitter reaches that, we will get some sort of deviation from the resting potential. So, and like, and the IPSPs are like when, like, you know, she used the example of hitting the knee, and then suppressing that. But any deviation from the resting potential, so the graded potentials are a means of reaching the threshold to activate the voltage gated. And once you activate the voltage gated, you're almost always going to get this exact reaction over here. So sodium, Na+, which is positive, enters into the cell, on the postsynaptic membrane. And once it enters, all the sodium is going to flood in. The millivolts is going to skyrocket. It gets super positive. Already in the cell was K+, all that potassium. They're both positive. So once you reach action potential, all the potassium is going to be like, yo, we can't handle that. So we're going to ball out of here. So all the potassium leaves, right? And so much of it leaves because the chemically gated potassium channels, that's key. The chemically gated potassium channels are open to allow that potassium to leave the cell. And so it's just going to flood out, right? And so we go below the resting membrane potential, kind of what it normally sits at. And that's where we get the after hyperpolarization. It goes way too low. And then it comes back up to resting potential. So a couple questions that she said that will be on the test. I'll talk through and kind of hopefully explain. So true, false, graded potentials are almost always caused by opening of chemically gated channels. That is true. After threshold, voltage gated channels are always open and we get the rising. So after threshold, so that's this line right here at negative 55 millivolts. After threshold, voltage gated channels always open and we get the rising to action potential. So this will always happen once threshold is reached. So once a graded potential, such as an EPSP or an IPSP, it happens, if it reaches threshold, this will always happen. And that is true, even though it says always. Okay, that was another question. All right, and then what is happening in repolarization? So this process of potassium leaving. Once the action potential is reached and sodium and potassium are present, potassium will leave the cell when the potassium voltage gated channels are opened, and potassium leaves at such a rate the cell is brought to hyperpolarization because they remain open past the point of resting membrane potential. So much potassium leaves that it goes below the resting membrane potential. Why does hyperpol? So that kind of goes to my next question. This was another one she included. She wanted to make sure we knew. Why does hyperpolarization occur? The voltage-gated potassium channels are open longer than to get to resting potential is all you really need to say for that. Let's see what else I have. I mean, that's hitting a lot of it. So, I mean, this slideshow, the synaptic transmission and integration. So, how do they travel between neuron to neuron or neuron to muscle cell is key. This is exactly how it will. Action Hillcock, it must reach there for that action potential to travel. Don't really understand what that means. I have it written down. Let's see what else. Oh, she wants us to know spatial summation and temporal summation. She said that was really good, really important thing. And those are just both types of synaptic integration. It's on the slideshow. I can pull it up on here too. Synaptic integration. There we go. That's these temporal and spatial. It's bottom left here. And basically, these are just ways in which EPSPs and IPSPs, SP, I don't know, I'm probably butchering it. These are just ways in which they can work to reach threshold or not reach threshold. So temporal, you'll see, is just a speedy firing of neurotransmitters to reach the chemically gated channels and to continually fire at a more rapid pace. Where spatial is you're sending multiple at the same time. She seemed really like, wow, the brain is amazing that it can do that. So I don't know why, but she really was. Anyway, so let me see if there's any other key things she said will be. So synaptic integration are the means by which threshold is reached or withheld. Or so, like, you know, synaptic integration can be a way in which it's like, well, we're going to keep it below threshold. We don't need a reaction here. So if like someone hits your knee, you don't need to kick your leg out. Action potentials always reach the same height. Potassium moving in and out of the cell. At peak, potassium flows out, opens the channels. Hyperpolarization. I think the one thing I definitely need to to study more is this first part of knowing, what's it called, how the neurotransmitter leaves its little block. Because it has something to do with calcium. It's important. It will definitely be on there. But I think that's it for that. And then the spider stuff is really going to be on there. So here's what you need to know for the point to ponder. Black widow spider venom. So when the black widow spider venom is present, it is going to cause no release of the neurotransmitter because calcium is going to be so it blocks. So there's going to be no release of the neurotransmitter because it blocks the release of the excitatory neurotransmitter. It won't be able to pass through that presynaptic membrane right here in slide 6 under point 6, 6C right here. Boom. They can't leave the neurotransmitter. I think can get out of the ball, but if you just say blocks the release of the neurotransmitter, because I need to study that more. But anyway, the consequence is going to be no action potential, no muscle contraction. The cobra venom, the neurotransmitter is released across the presynaptic membrane, but the neurotransmitter cannot bind because the cobra venom blocks the chemically gated channel here, and so it can't pass through. So that's the cobra. Neurotransmitter can pass, cannot bind to the chemically gated channel. Therefore, the voltage gated channels cannot open. And so there's no action potential reached. And therefore, the results are the same, that there's no action potential and therefore no muscle contraction. Why are muscle contractions important? Because it makes our diaphragm move, which allows us to breathe, and our heart to pump blood. All right, I mean, that's a lot of what we talked about tonight. I try to regurgitate as much as I could. I'll send you pictures just because someone just sent me a picture of notes about this stuff. I'd be like, what the frick is that talking about? I hope that helped make sense. It's all about, so here's the neurons. from the brain all the way down through your central nervous system to your peripheral